Hello everyone, today I will present about water quality control that I attach at Pejabat Kesehatan Daerah Hospital under Practical 3 of Food Safety and Water Quality. I am Koyy Binti Panus, Bachelor Environmental Health and Safety Honors student. In this presentation, I will explain the importance of water quality control, sampling procedure, microbiological and chemical sampling, equipment calibration, documentation, quality assurance, and reporting. Water quality control is important because drinking water must be safe for the community. The Kemam unit monitor drinking water quality to detect contamination earlier and reduce the risk of water-related disease in Sipitang. This activity also help protect public health and support cooperation with local water authority to maintain a safe water supply. And the objective of this presentation are to explain the role of Unit Kemam in water quality control and describe how sampling schedule are prepared. I will also explain the difference between microbiological and chemical sampling, identify the equipment and calibration requirement, and finally discuss quality assurance reporting and regulatory control. Okay, before conducting water sampling, a proper sampling schedule must be prepared. First, we identify the sampling locations such as the treatment plant, reservoir, water mains, public premises, and consumer taps. The frequency of sampling can be daily, weekly, monthly, or based on the risk and previous result. The team date, transportation, cooler box, ice pack, label, and forms must also prepare. A good sampling schedule help ensure the sample are representative and delivered properly to the laboratory. Okay, this slide show the basic workflow for sampling. For water sampling, first we prepare the equipment and verify the sampling site, and next we flush the tap or water source before collecting the sample. And after collecting, the sample must be labeled and all info are recorded. And the sample is then stored. The sample is then stored properly in the cooler box and transported to the laboratory. The main principle is to avoid contamination, maintain traceability, and preserve the condition of the sample until analyzed. For chemical sampling, microbiological sampling. Microbiological sampling is carried out to detect bacteria and assess the risk of waterborne disease. It is used to detect organisms such as Total-coliform and E. coli. A sterile bottle is used and sodium thiosulfate may be added in the water is chlorinated. Before collecting the sample, the tap should be flushed for about 2 to 3 minutes. We must avoid touching the bottle mouth and cap to prevent contamination. And after the collection, the sample is labeled, placed in the cooler box at about 4 degrees Celsius, recorded in the Kemam A1 form and logbook, and sent to the laboratory within the required holding time. For chemical sampling, is carried out to measure parameters such as pH, turbidity, chemical metals, and other chemical substance. The correct sample bottle must be used depending on the parameter. For some parameter, preservative may be required. For example, a nitric acid may be used for trace metal sample. For pH and turbidity, preservation is usually not required. The site condition, temperature, and fuel reading should be recorded in the Kemam Form 2. The sample are then transported using a cooler box and submitted to the laboratory according to the required procedure. For equipment and calibration, reliable water quality results depend on properly calibrated equipment. For example, a pH meter is calibrated using standard buffer solution such as a pH 4, 7, and 10. The turbidity meters use standard solution while the chlorine meter is used to measure the chlorine residual. And the other equipment include the conductivity meter, dissolved oxygen meter, and microbiological testing kit. All equipment should be calibrated before use. Risk between sample and the calibration result should be recorded in the logbook. For form, label, and chain of custody, proper documentation is very important to prevent sample mix-up. The required document include a CMAM 1, CMAM 2 forms, analysis request form, bottle label and sample ID, field logbook, and like that I said before, chain of custody recorder. The important information, the good documentation ensure that every sample can be properly traced from the sampling site until the laboratory. For quality assurance and reporting, quality assurance and quality control are important to ensure that the results are accurate and reliable. This include using certified bottle, blanks or duplicate sample, calibrate equipment, and train staff. After laboratory analysis, the results are reviewed to identify any deviation from the required standard. If contamination or non-compliance is detected, corrective action must be taken quickly. Overall, a good water quality control depends on proper sampling, correct equipment use, and accurate documentation and timely action. Okay, these are a few one of the sampling point that I going to take the sampling. This one is Jabatan Air Sabah Main, and before we take the sampling, we must record the well pack, record the date, and what sampling, what sampling that you do, like otherwise the microbiological or chemical. Here we're doing some sampling to detect the pH, reading the pH, turbidity, and NTU. And in conclusion, water quality control is an important environmental health activity because it helps protect the community from unsafe drinking water. Sampling must be properly planned according to suitable location, frequency, and parameters. Microbiological and chemical sampling require different bottle handling and transportation methods. Calibration, quality assurance, and complete record are also essential to produce reliable results. If the results are not compliant, reporting and corrective action must be taken according to procedure. And that's all my presentation, and thank you.